Today, we're going to talk about a molecule that completely blurs the line between our own brain chemistry and the wild world of psychedelics. It's time to rethink DMT. All right, let's just jump straight into the deep end here. Imagine this. What if one of the most powerful psychedelic compounds on the planet wasn't really a drug you take, but something your own brain is making and using, like right now? Because that's exactly what the science is telling us. We're about to find a molecule we all think we know, DMT, but in a place most of us would never expect to find it, inside our own heads. Yeah, so when most people hear the letters DMT, your mind probably jumps straight to those really intense, super fast-acting psychedelic experiences, right? For as long as we can remember, it's been filed under external drug, something you put into your body from the outside. Okay, but here is the plot twist that changes the entire story. A huge wave of new research is basically confirming that DMT isn't some foreign invader at all, it's endogenous. And that's just a fancy way of saying it's homegrown, made right there inside the mammalian brain. Yep, that includes ours. And get this, we're not just talking about tiny, insignificant traces. I mean, when scientists literally sampled the fluid directly from a living rat's brain, what they found was kind of shocking. The levels of DMT were right up there with serotonin and dopamine, the big-name neurotransmitters that run our mood and motivation. So this isn't some minor detail. DMT is a serious player in the brain's chemical soup. So that obviously begs the huge question, right? If our brain is cooking up its own potent psychedelic, what in the world is it doing with it? Let's follow the clues. So if we start treating DMT like any other neurotransmitter, we've got to ask, what's its job? And it turns out the first big clues about its, 9 to 5 function, they pop up when the brain is really under pressure. This study from way back in 1981 just paints a perfect picture. Look at this chart. Researchers checked the DMT levels in rats that were happy and housed in a group versus rats that were stressed out from being housed alone. In the normal, happy rats, DMT was basically zero, undetectable. But in the stressed out rats, whoa. The levels just went through the roof. And we're not talking a little bump. This is a massive, dramatic response to stress. So what does that tell us? It tells us that DMT isn't just hanging out in the brain doing nothing. It's part of an active, adaptive system. It's like the brain hits a panic button during stress and starts cranking out DMT, maybe as a form of neuroprotection, almost like a chemical bodyguard for our brain cells. Okay, clue number 2. This one takes us all the way back to the beginning of life. Another study found DMT in the brains of tiny developing rat pups, and the levels weren't flat. They actually peaked around day 17, which is a super critical window for brain development. So you have to wonder, why would it be there during that massive construction project? Well, it points pretty strongly to a role in neuroplasticity. The brain's amazing ability to grow, to build new connections, to literally rewire itself. That's the bedrock of all learning and memory. So we've got 2 huge clues now. A stress shield and a brain build. But to really get how DMT could be doing all this, we need to know how it actually talks to brain cells. And this is where the story we've been told for years starts to completely unravel. It's pretty cool. Okay, so for decades, the classic theory for how psychedelics work was actually pretty straightforward. A molecule like DMT floats up to a neuron and it plugs into a specific little docking port on the outside of the cell. This port is called the serotonin 2A receptor, or 5-HT2A for short. That connection is supposed to kick off the whole trip. That's been Psychedelic Science 101 for a long, long time. Well, until now, a crazy paper from 2023 just dropped this bomb on the whole field. When they looked for this exact thing happening in living animals, they found, and I'm quoting here, completely lacking evidence for significant occupancy by DMT at 5-HT2A receptors. Just let that sink in for a second. They pumped the brain full of DMT at levels almost 10 times higher than it should have taken to stick to those outside receptors. And crickets. Nothing happened. The receptors were empty. The main suspect, the surface 5-HT2A receptor, had a perfect alibi. The classic theory was suddenly in huge trouble. So if the party isn't happening on the outside of the cell, where is it? Well, it turns out the answer was hiding in plain sight the whole time, just inside the building. Now to get this, we just need a tiny bit of chemistry. Think of the wall of a grain cell like a bouncer at an exclusive club. Serotonin, well, it's a polar molecule. It can't get past the fatty velvet rope of the cell membrane. It's stuck outside. But DMT?
DMT is different. It's lipophilic. It loves fat. So it just waltzes right past the bouncer, through the wall, like it has an all-access backstage pass. And that gives us the new theory, step by step. Okay, step 1. DMT uses that all-access pass to slip right through the cell membrane. Step 2, it totally ignores the receptors on the surface that everyone was looking at. And then step 3, this is the big one, it finds and binds to a completely different set of those same 5-HT2A receptors, but these ones are inside the cell, hanging out near a structure called the Golgi apparatus. Think of the Golgi as the cell's main factory and shipping department. This is a total game changer. It suggests there's this secret internal communication system that the main player, serotonin, can't even get to. And DMT isn't just one of the keys for this internal lock. It might be the brain's very own custom-made key designed specifically for this hidden pathway.
So what does all this add up to? Well, finding this internal system doesn't just make us rethink psychedelics. It makes us rethink how the brain itself is wired. And its location, near the Golgi, is not random. That's the nerve center for all the stuff we were just talking about. Neuroplasticity, building the cell's wiring, the axons and dendrites, repairing synapses, and even protecting the neuron from damage. This internal DMT system is right at the heart of brain maintenance and growth. And suddenly, it all clicks into place. This new mechanism could be why psychedelics seem to have so much therapeutic potential for regrowing neural connections. But maybe, even more importantly, it could explain the everyday, fundamental job of the DMT our brain is already making, to be the background maintenance crew, constantly repairing, protecting, and upgrading our neural hardware. So here's the big takeaway. We're not just looking at some quirky molecule the brain happens to produce. We might be looking at a whole dedicated network that some researchers are now calling an endogenous hallucinogen neuronal system, a built-in system that uses a psychedelic molecule for the most essential jobs: protection, repair, and growth. And that just leaves us with this one final mind-bending thought. The question isn't if our brain makes its own psychedelic anymore. The question is why. We have a system, built into us, that uses a reality-bending molecule for basic upkeep. So what does that say about the nature of consciousness itself, and where we draw the line between the world inside our heads and the world out there?